So you have all this information in your procedures, and you especially have a handout called the published article. So I urge you to read that published article and really try to understand the steps, which is really, I'm going to start with that. I'm going to explain what the purpose and the chemistry behind the experiment here. And in this presentation, I have detailed the exact volumes of each of your steps that you should use. So take your slides, bring them into the lab, and this will, you won't make any mistakes if you can follow what I'm going to explain here. Okay, so the purpose of this experiment is to give you some real experience doing strain development, which is what happens in many biotechnology companies, whether pharmaceutical or biotech companies. The chemical engineers don't usually do the molecular biology. But when you do it with your hands and you try to make it work and you read those procedures and you think about it, you're going to be much better prepared to dialogue with the people who are working in the biology department. Okay? Your job would probably be to do the scale-up of the strain. But they may be handing you new strains every day, and you don't want to just be growing up whatever it is. You want to know what they did. You want to understand what they did. So the purpose of this experiment is to give you some introduction into gene subcloning, so that you can understand at least some of what the people in the biology group are doing in those companies, okay, that you will be working with. So a subcloning is when you already have the gene not in the native organism, but it's already on another plasmid. Okay, when you already have it on a plasmid, you know the sequence, you know everything, it's easy, much easier than starting with an organism where you got it. Take the organism, make a cDNA library, and then you do PCR to try to find the gene. And then when you find the gene, then you need to amplify that piece, and then you want to try to figure out how to cut it, you sequence it. It's very complicated. In our case, we're just going to do a subcloning, which means that we've already got that GFP gene on a plasmid. All right? So that'll make it a little easier. Let's look at the gene. This first gene is the gene where we are taking the green fluorescent protein gene from. All right? This is called the P-GLO plasmid. And this P-GLO plasmid has some interesting features for us, and I will show you. This is a little bit of a review from your biochemistry or Chem 192 class that should have been the preparation for you to take this class. Okay, first of all, we have an E. coli origin of replication. What is an origin of replication? That is the sequence that tells the DNA replication to start. So if you've got that on the chromosomes, so when your cells divide, they make the new DNA. And the bacteria do the same thing. They make a new chromosome. But if this plasmid is in the bug at the time of replication, it's going to make more of this too, because it's got the origin of replication. Right? Then we have the green fluorescent protein gene, which is the one we want. We want to take that gene and put it on another plasmid that has higher expression levels. All right. Then this plasmid has the arabinose promoter. Okay, you remember what induction is? When you, if you have an inducer, in order for the polymerase to bind, the inducer has to be present for that particular genetic system, right? So in the case of this plasmid, it is the arabinose promoter. So when you're growing a bug and you want to express when you want to express the GFP gene with the P-GLO plasmid, then you will use arabinose to induce the expression. With our PET plasmid, we induce the expression with lactose or IPTG. And I'll explain later why we don't need either of those, because it's some things in the LB broth that does the induction, so we don't really need it. But anyway, okay, the last thing on here is BLA. Okay, the beta-lactamase gene. This gene product confers resistance to ampicillin. So that means if this plasmid is in the organism, that you can feed ampicillin, which normally would kill the organism. You can feed it to that organism and it won't die, because this plasmid is giving it a gene that will make the organism resistant. So it'll grow right in the midst of that ampicillin. But that's great, because then every organism in that soup is going to die except for the one that has the plasmid in it. So we can select for the bugs that have the plasmid in it. Okay, so this is a complete plasmid for expressing green fluorescent protein. Let's look at our new plasmid. Our new plasmid is the PET plasmid. This plasmid does not have ampicillin resistance. conferred by it. It has kanamycin. So it has a gene that confers resistance to kanamycin. And it also has the E. coli origin of replication, okay? And it also has a multi-cloning site. So multi-cloning sites are closely spaced, popular restriction enzymes, cutting sites. So if you can use any one of those restriction enzymes, then you can cut and put your gene. So that makes it much easier, all right? So in fact, let's take a little closer look at this little region here. This is the part that normally you would somehow put your gene in the middle here. And let's see if we can do that, all right? So this is the multi-cloning site. This shows you we have a T7 promoter. What is a T7 promoter? A T7 promoter is a bacteriophage promoter. So you know what that is. It's a virus that attacks the bacteria. And it attacks them. It'll use their genetic machinery to make its own DNA. And then it can express its own proteins. Okay, so the phage needs the cell, it needs the cellular machinery for its own replication. So it's not alive. That's what we learned about viruses, right? Okay, so it's got the T7 promoter, so it's a very strong promoter. It's stronger than the arabinose promoter, all right? And it also has the KPN1 restriction enzyme. That's going to be our downstream restriction enzyme. Okay, so we are going to put our gene in right here between the XPA1 and the KPN1 site. So we're going to take this plasmid